Welcome back. In this last video on nuclear and cell division, I'd like to take a look at the process of cytokinesis. So after the chromosomes are separated and they've been segregated to each end of the cell, cytokinesis divides the cytoplasm into two to give us two new cells when the membrane pinches the cell into two. So the process starts in late anaphase or early telophase, but it's considered to not be a part of mitosis. So just to review this again, there's M phase, and that is mitosis plus then cytokinesis. So in animal cells, cytoplasmic division is called cleavage. It begins with a slight puckering of the cell surface that deepens into a structure called the cleavage furrow. And then the furrow deepens until opposite surfaces of the cell make contact. So if you think about this in cross-section, if this is the cell, then there's going to be this puckering and deepening of the furrow until eventually the two membranes, think about it in 3 dimensions, are gonna make contact. And so that happens along a plane that passes through the middle of the spindle where the metaphase plate was, called the spindle equator. The actual cleavage event depends on the structure called the contractile ring, and that's a ring of actin that works with a myosin protein. We'll take a look at it in a moment. And so that ring of actin is just below the just below the membrane, and as the cytokinesis proceeds, as cleavage proceeds, then the ring tightens around the cytoplasm to effectively pinch this one elongated cell into two daughter cells. Contraction of that ring. So here we're seeing a cell which is just forming its cleavage furrow. Contraction of the ring is generated by interactions between the actin filaments in the contractile ring and filaments and members of a non-muscle myosin 2. There's a motor protein like the one we saw in muscle contraction, but this is a non-muscle myosin called non-muscle myosin 2. It's also dependent on members of the Rho GTP binding proteins, and they regulate the assembly and activation of the contractile ring. So we talked about Rho-RAC CDC42 when we talked about changes to the cytoskeleton. So here is one of those, more specifically Rho-A, being recruited. Down here you can see that Rho is being recruited to the side of the cleavage furrow where it activates proteins needed for the polymerization of actin, and it stimulates the switching on or the activation of myosin. And so you can see the Rho localized to the cleavage furrow. So as cytokinesis ends, the connection between the two cells narrows down to like a little thin stalk between the cells, and as cytokinesis really completes, you end up with a structure called a midbody, which is an electron-dense area with a lot of tightly packed microtubules, which are remnants from the spindle. And so that persists until the process is complete and we have abscission, which is the final separation of the two daughter cells. Now, what we've seen so far really is symmetrical cell division, so cleavage down the middle of the cell, but that's not always the case. Sometimes the spindle forms in an asymmetric fashion, resulting in the cleavage furrow forming in an asymmetric place. And so that can result in one cell that is larger than other cells. So you can see this cleavage furrow forming here, dividing the cell in two, but then at some point after 2 or 3 divisions, the frog embryo here, which is developing into a tadpole, the frog embryo here, you can see that these cells up here have divided unequally from the cell down here, and so we've got a large cell accompanied by smaller cells. So this is not uncommon during embryonic development when you are trying to give cells different developmental fates, if you like. So this cell could well differentiate into a different cell type than this one down here. And so that asymmetric division can contribute to varying developmental potentials. And you can see down here in this sea urchin embryo how asymmetric the spindle is. And so the cleavage furrow will form here to give you a bigger cell up here and a smaller cell down here. So that's wrapping up the end of nuclear and cell division 3. Next time we'll start taking a look at how all these processes are regulated. So we'll have several videos on the regulation of the cell cycle, and I'll see you for that very soon. Bye for now.